A very good morning to all. In the last class, we discussed the type Chlamydamonas coming under chlorophyceae, and you studied the structure of Chlamydamonas and also how it reproduces asexually. Okay, so we are moving to the second part of that reproduction, that is sexual reproduction in Chlamydamonas. So this sexual reproduction is taking place in unfavorable conditions, and in Chlamydamonas there are three types of sexual reproduction. What are they? Isogamy, anisogamy and oogamy. So you can from the term itself, isogamy, iso means similar, aniso dissimilar, oogamy means oogonia or egg is participating. So these are the three different types of reproduction, sexual reproduction occurring in Chlamydamonas. Okay, so what do you mean by isogamy? First one is isogamy. So isogamy is the fusion of similar gametes, that is morphologically similar gametes fuse, but they differ physiologically. Okay, so they produces gametes of one type, same type, and they undergo fusion. So I told you there is physiologically different. So you can represent them as plus strain and minus strain. We usually term it as male gamete and female gamete, but it is morphologically similar but some physiological differences. Then you can name it as plus strain and minus strain. Okay. So gametic union occurs. How these gametes are formed? So any vegetative cell, any Chlamydamonas cell, you know, that is unicellular, can act as a gamete. So before gamete fusion, each cell that undergo division and produces up to 16 or 32 gametes, and all the gametes are biflagellate, means two flagella. See, this is the fusion, isogamete, similar gametes. So shape and morphology same, but this marking because it is physiologically both are different. Clear? See how it is formed. The single cell acts as the gamete, and before producing numerous gametes, its protoplasm undergo repeated divisions, mitotic divisions, okay, and form 16, 32 and 64 cells. They are modified and become gametes. They are unicellular, uninucleate and biflagellate. Contain single cell, single nucleus and two flagella, each gamete. Okay. Now, so gametes fusion, how it take place? Fused by anterior end. You know the one pointed end for Chlamydamonas, shaped like this. Pointed end, that is anterior end. The two gametes, two anterior ends fuse. Okay. Then two flagellate, 2 plus 2, 4 flagella coming together at its anterior end. Okay. Then fusion of nucleus and fusion of protoplasm of the two. That is, fusion of nucleus is karyogamy and fusion of protoplasm is plasmogamy. These two events take place and a quadriflagellate zygote is formed. So the fusion product is known as what? Zygote, and it is quadriflagellate, means 4 flagellate. Okay. Now this zygote moves sometime, motile sometime, they later come on to rest. Okay, and this produces thick wall around the zygote, and now you can call it as a zygospore. Clear? So fusion at anterior end, it become quadriflagellate structure, fusion of nucleus, fusion of protoplasm, then flagella become withdrawn or lost. It produces a thick covering around it. Now you can call it as zygospore. Okay. See how it is developing. So first Chlamydamonas cells produces gametes here, male gamete, female gametes undergo fusion, two flagella of one and two flagella of other, to quadriflagellate fusion, mixing up of nucleus and protoplasm, quadriflagellate structure, fusion product, that is plasmogamy, karyogamy, product is zygote. It produces thick wall. You can see sculptured, designed wall around the structure, that is zygospore. See, this is n number of n plus n number of chromosome here also. So n plus n fused and diploid condition 2n reaches. Now it undergo meiotic division. You know what is meiosis? A diploid cell undergo meiosis and produce 4 haploid cell. So likewise it undergo division and 4 haploid cells produced. 2 become plus strain and 2 become minus strain. Clear? So plus strain and minus strain. The plus strain produces gametes, minus strain produces gametes. Or like zoospores, they may be released as zoospore or they may produce as gametes, and life cycle continues. That is isogamy. Okay, now second one is anisogamy. What is the difference? The term itself indicates two morphologically dissimilar gametes undergo fusion. Okay, it is first reported in Chlamydamonas brownie, and gametes formed by division of protoplast. Gametes formed by the division of protoplast. Okay, so larger gametes are formed by are called macrogametes, and they are the female gamete, and smaller one is the microgamete, or they are the male gamete. So size difference is there, that is anisogamy. Okay, now this is two morphologically dissimilar ones, one is small, one is large. Then these gametes liberated and undergo rest, they fuse at the anterior end. See the difference here. Contents of male gamete drift into female gamete. But what happens in isogamy? Both mix together. Here male gametes contents move into female gametes. Clear? Then flagella withdrawn, fusion take place, and diploid zygote is formed. Then the development of after zygote is same as that of isogamy. Okay. Now what is oogamy? Oogamy is first reported in Chlamydamonas coccifera. So it is any vegetative cell withdraw flagella, come to rest, enlarge in size and function as an egg. Clear? A vegetative cell withdraw flagella, enlarge in size and function as an egg. Okay. Or that is oogonium or egg. Then number of small male gametes produced, they are also called microgamete, male gamete or androzoids. They are flagellated one. This is not a flagellated one. Okay. They are spherical, uninucleate, biflagellated one. Okay. Now male gamete swims, moves and reaches to female gamete, that is what non-motile, and unites at its anterior end. Then usual case plasmogamy, karyogamy and zygote is formed. So this is condition. Here the oogamy is most advanced sexual reproduction. Here what happens is the vegetative cell withdraw flagella, enlarge in size and function as macrogamete or egg, and that become that undergo rest. Then male gametes are very small, microgamete are small, they are also known as androzoids, they are biflagellate, they are fast moving, and they reaches to female gamete or egg, fusion take place and rounded off and zygote is formed. Okay. See, this is oogamy. Male gamete only flagellate and it's fused with a female gamete or egg. Now, what are the conditions of zygote? How zygote develops? Zygote is motile for some time, then it undergoes rest, settles on a substratum, on a space. Then the zygote is filled with nutrient-rich food, usually oil and starch. Then zygote is also called zygospore. When it undergoes a thick water cover produced it undergoes resting period. Okay. Now germination. I already told the gametes are haploid, so they undergo fusion. So zygote is diploid. So diploid nucleus undergo meiotic division and give rise to 4 haploid nuclei. Diploid nuclei undergo meiotic division and produce 4 haploid nuclei. Okay, which form 4 haploid zoospore-like motile cells. It undergo division and forms zoospores. Okay, zoospores liberated and into new Chlamydamonas cell. This is a condition or development in isogamy, anisogamy and oogamy. After zygote formation, this usually zygote undergo this process. Clear? Now, see here, this is egg gametes fusion. How androzoids formed? One cell acts as androidium, protoplast divides many cells, each become biflagellate androzoids. This is oogonium. That oogonium here, no flagella, enlarged in size, egg or oogonia. Androzoid attach its anterior end, withdraw flagella, contents move into its 2 nuclei, one nucleus of oogonium, other nucleus of androzoid, come together, fusion take place, and this is a zygote. Clear? Okay. This is a Chlamydamonas life cycle, asexual reproduction, sexual reproduction. So how can you differentiate isogamy, anisogamy and oogamy? In isogamy, fusion of similar gametes, they are morphologically similar, physiologically dissimilar, both are motile. Clear? Same size. Okay. Then fusion takes place, zygote develop into zygospore, then liberate as zoospore. Okay. Now, anisogamy, morphologically dissimilar. One is small, other is large. The content of male gamete drift into female gamete. That is the point here. Then you shall develop into zygote, zygospore, zoospore, Chlamydamonas. Okay. Then what about oogamy? One of them withdraw its flagella, one vegetative cell, enlarge in size and function as egg or oogonia. Okay. Then number of small male gametes develops from another cell, that is antheridium, produce antherozoids. Antherozoids are biflagellate structure. They move and they attaches to the oogonia. Oogonia come to rest on a substratum. Antherozoid attaches its anterior end, flagella withdrawn, contents mixes together and zygote formed, then zygospore, then zoospore, and finally Chlamydamonas cell. So that's all about the sexual reproduction in Chlamydamonas. So these questions may arise for essay type. Okay. So now you can have a clear session to clear your doubts. So how Chlamydamonas reproduces actually, what are the three types, how can you differentiate each, and also some examples. Okay. I hope you enjoyed well. Thank you.